Hello, Anna here, and I'm going to go over part 6 on the excitation coupling and how you're organizing stuff inside the myofibril organelle. So let's get to it. All right, so in this section, I have one major notes slide right here where I've got my structures, what they do, all the things about them, all kind of organized into one space. So I'm not going to read this to you, but you have the notes on it. What we're going to do next is kind of go through the pictures. All right, here we are on the first picture. And if you remember from previous sections, we've got the plasma membrane, which can also be called the sarcolemma. Okay, what I like about this particular version or this particular drawing is that they put in the holes for the T-tubules, where the T-tubules are going to invaginate. All right, the T-tubules are being represented in green along here, okay? So each one of these little holes is coming down to a T-tubule. Now this is important because this is how the action, the action potential gets down to the sarcomeres. All right, we need a pathway for that to occur, okay? So the plasma membrane from the neuromuscular junction is going to travel along the along the sarcolemma, so it's going to ripple out. So it's kind of going to go in rings like this and travel out concentrically, okay? So that would be like the action potential. It's a terrible drawing, but anyway, you get the idea. Now, for these T-tubules, they go down to a sarcomere. So we've got the plasma membrane, and here, all of these structures right here are the myofibrils. And then we've pulled a myofibril out, and we're going to blow it up to look at it. All right, so this structure right here is being shown right there so that you can see that this is the myofibril, which is a chain of sarcomeres. And you've got all of these structures here coming down. Now, what I need you to remember before I go on to the next slide, I need you to remember this for the next slide because you won't see it, is how this plasma membrane has the holes that go to your T-tubules, okay? So let's flip over to the other slide so we can see this a little bigger. All right. So we're looking at this a little bigger. And what you're going to have is we're just going to pretend that there is a plasma membrane up here, and it's going to invaginate. Plasma membrane up here, it invaginates. And when I have an electrical signal, it's going to travel along here, and it's going to go down here. Now, as it's going, it's depolarizing the membrane, okay? That depolarization is going to go down here, all right, and then travel around here, travel around the ring of the T-tubule. As it does that, it's going to open voltage gates for calcium that are in the sarcoplasmic reticulum. Okay, so the sarcoplasmic reticulum, the SR, stores and releases calcium. All right, calcium is toxic if left loose. So we need to sequester it so that it doesn't cause the other structures inside your cell to start going through necrosis or apoptosis. So it's all being stored in these blue lines. All right, so when an action potential travels down here, all right, goes down the T-tubule that opens up the voltage gates of the sarcoplasmic reticulum, calcium then floods into this space all around here. So that's really bad representations of calcium. Here, we'll do this. So we got calcium molecules floating all in here. And remember the calcium, all right, so if this is calcium, it's then going to go to your sliding filament mechanism. And in the sliding filament mechanism, it will move the troponin and tropomyosin, which will then unblock the G-actin attachment site, and then your myosin crosshead attachment can occur. Okay? All right, let's look at the next slide. All right, on this slide, we've got a blow up, and I really like this picture because we get this great view of our plasma membrane, which in the muscle we're going to call the sarcolemma. Okay? And what we're seeing are these paired gates, and these are voltage gates. And in this case, these are voltage gates for sodium and potassium, okay? And what you'll notice is that going all along here, I've got paired voltage gates going along the sarcolemma and then down into the T-tubule. So this section is the T-tubule, which is going to go down and wrap around the sarcomeres. Okay, let's look at another slide. All right, in this picture, they're kind of putting it all together. So you've got your neuromuscular junction here. So I would have an action potential that would come along here. It would go down in here. Here it will trigger calcium to come in through calcium voltage gates. And then the calcium is going to trigger this to go here and via exocytosis enter the synaptic cleft. Then that binds to these chemical gates for sodium. Okay? That opens it up, and then sodium can enter into this area. The sodium, in turn, is going to change the polarity. So it's going to start depolarizing the membrane. If it is enough depolarization that the change makes it over here, where I have voltage gates for sodium. If it makes it there, then it will depolarize and generate an action potential. Okay? So now I've got an action potential running along here. Let's zoom this in. Whoops, I need this button. It's going to run down the T-tubule in green. Okay? Now, in here, they're not showing it, but I have what I'm going to call voltage gates for calcium on the sarcoplasmic reticulum membrane. Okay? When I change the voltage here, it also changes the voltage there, and that will allow calcium to leave and drift out all along here. And remember, this is where you have your sliding filament mechanism with the myosin and the actin. So here you can see they're showing the calcium here, okay, and seeing how it's drifting all over the place, okay? What have I done with this? Okay, now that brings us down to this structure, which is again the sliding filament mechanism. Okay, and so now you can see these are those calcium molecules, which are then going to come over here, all right, and move the troponin, all right, move the troponin so that this can attach to that and you get the crossbridge attachment. Okay, so that's the crossbridge cycling right there. All right, let's look at the next slide. All right, now we're going to do this in detail and we're going to back it up and look at it closer. All right, so we're going to zoom in here. I have an action potential moving down my axon. All right, that is depolarizing the sarcolemma here. Okay, that is going to come over here and look at this. I love this picture. It's one of my favorite pictures because it shows the voltage gates for calcium. When I depolarize this membrane right here, it will open these gates, allowing the calcium to come in. The calcium comes in and attaches to the vesicles. That triggers the vesicles to come over and merge with the plasma membrane down here. When that happens, you can then do exocytosis and the neurotransmitter can come out. In this case, this neurotransmitter that we're dealing with is acetylcholine. The acetylcholine comes here to my chemical gates for sodium, okay? The chemical gates open, okay? And now sodium molecules can flow into the sarcolemma, into the sarcoplasm. All right, let's look at the next slide. Actually, before we go on, I just want to bring up one more thing just in case it's not on a later slide. ACHAs, acetylcholinesterase, is always present. All right, so basically it's always going to be going around trying to find the acetylcholines, and it's going to start breaking them down. So as soon as it starts being released, the acetylcholinesterase is trying to destroy it, okay, to basically shut down the transmission. And that begins as soon as you start releasing acetylcholine, okay? All right, now we're going to go on to the next slide. All right, now on this slide, they're not showing some of the other things, but what they are showing now is you can see the sodium a little bit better, and you can see how you open up this gate with the acetylcholine, and now these sodium molecules are going to go in. I also have potassium molecules. The potassium molecules can follow their concentration gradients, but really what is driving this part is going to be the flow of sodium. Okay, that's the primary driver of this section of the action potential beginning. Okay, this is actually a graded potential at the moment, but anyway, what thing I want you to focus on are the colors right here of these gates. The reason I want you to focus on that is because when you get over here, I want you to notice that they go to lavender and pale blue instead of gray. Okay, so all right, so the gray, so gray equals chemical gate, and then the lavender and blue are voltage gates. All right, if I do not get enough sodium to come in through the chemical gates to change the polarity here, to depolarize this section, there will be no action potential, okay? I will only get an action potential to generate if it's enough of a voltage change here to trigger that to open, okay? If this sodium gate opens, then tons and tons of sodium come in. And this is a self-propagating, all or nothing thing. Action potentials, if they start, they're going to go, and they're going to go forever, okay, until they hit the end, okay? Now, these are a pair. I mean, they are separate, but they function as a pair, so that I've got the sodium gate opening to let sodium in, and then the potassium gate to allow potassium out. This part is responsible for depolarizing. This part is responsible for repolarizing, okay? When I get an action potential, it's basically going to start here, and then it's going to go down this way, okay? But as this section is depolarizing, this section is beginning to repolarize. So that I repolarize, so that I depolarize, and then I repolarize. Depolarize, repolarize. So it does this pattern, this wave-like pattern as it goes along. Okay? All right, let's look at the next slide. All right, here we're looking at a blow-up of the T-tubule. So we see the sarcolemma here. We've got our paired sodium and potassium voltage gates. The action potentials traveling along here, opening all of these up, including all of the gates down in the T-tubule, okay? That causes this area in here to change its charge. Enough that it depolarizes the next segment down here, but it also depolarizes these. These are my voltage, let's change that color, that's hard to read. Voltage gates for calcium that are located on the sarcoplasmic reticulum. So this is the sarcoplasmic reticulum. So these open in response to this, and then the calcium that's in here flows out into the sarcoplasm. So you can see I've got all kinds of calcium coming out here, okay? All right, let's look at the next slide. Now, when all of that happens, now you can get your crossbridge cycling and your sliding filament mechanism to work, okay? So right here, they're showing you a sarcomere, okay? And then here, we've got our actin, we have our tropomyosin. Oops, I'm misspelling that, tropomyosin. And we have our troponin, okay? Down here is the myosin, okay, our double head right there. I want you to pay attention to the ADP and the P molecules that are still attached to it, okay? Now, the next step, what are they doing here? Anyway, we're going to come over here. So with the calcium molecules, so I've got calcium molecules, they're going to come over and they're going to move that troponin and the tropomyosin, and this allows the myosin head to attach to the actin, okay? Now, as soon as it attaches, and you can kind of think of it as like a magnetic force, okay? As soon as it attaches, it then triggers a pivot, okay? Whoops, let me move this. So now we've got the power stroke or pivot. So this goes forward, all right? And that moves everything towards the center of the sarcomere, okay? As you're doing that, you lose the ADP and the P molecule, okay? Now, this is stuck here until you get an ATP molecule. So right here, I like the way they're showing the shadow right there versus the real thing. So we've got the shadow here. The ATP molecule attaches to this, actually attaches right here. So you've got an ATP molecule and it attached to the shadow. When it attaches to the shadow, this pops off. Okay, so now it's detached, but its head is still in an uncocked position. So the attachment sites are in this direction, and they need to be over here in order to attach to another one of those. So you need to recock this thing. And you're going to do that, and they're not showing that here. I'll just write it in. Hydrolyze the ATP to cock the head. So you go from here to here by hydrolyzing the ATP molecule. That causes the shadow to pop up into a position where it can be used again. And now you have, again, an ADP and a P molecule attached to the myosin head. Okay, you want to draw this out many, many times. Okay, next slide. All right, so what we're going to do is we're going to draw this all out, okay? And this is what I would do on the board if we were in class. So I have up here an axon, and this would be my axonal terminal. Okay, and then over here, I'm going to draw my sarcolemma. Okay. What I want to remember is that I have 3 sodiums on the outside for every 2 potassiums on the inside. And what that means is that I have a negative 70 millivolt polarity, okay? And this is what we call resting membrane potential, okay? Actually, I was just looking at neurophysiology, and so that's why I'm thinking negative 70. Negative 90 for our muscle cells, okay? So what's going to happen is I'm going to have an action potential that comes down. It's annoying. Okay, hold on. An action potential, which is your electrical signal traveling down my axon. It's going to come over here where it's going to open a voltage gate for calcium, okay? The calcium, which I'm going to represent as red dots, floats in. And then in blue, I have vesicles that hold acetylcholine. These vesicles come up to the surface and they do exocytosis. And then I've got, oh, I was doing that in blue. I guess I should be consistent. In blue, acetylcholine neurotransmitter. Now here, in red, I'm going to show that I've got chemical gates for sodium, okay? The acetylcholine is going to come over here, and we're going to show that the acetylcholine binds to the gates, and that opens the gate, okay? Now, I can take sodium molecules, and they can float down into here, all right? Because they are positively charged, that's going to take the negative 90 and make it less negative. All right, if it reaches threshold, all right, so remember we've got this little graph right here, and I showed how we have negative 90, then we might have negative 65. Negative 65 is a threshold. This is where the resting membrane potential is. If it gets to there, no action potential. If it gets to there, no action potential. But if it gets up to here, this is my action potential. I need to change the polarity enough that I can reach threshold, and then it'll repolarize and come back down, okay? So if I get enough of a charge change that I get over to this area, all right, so this is the, oh, I need a new color, motor end plate area. All right, if I get over here to the rest of the sarcolemma, let's use a yellow, and this is going to be a sodium voltage gate. And then in green, we're going to put in a potassium voltage gate, okay? If I get this to here and those voltage gates open these so that sodium can come in and potassium, actually potassium is going to go out. The sodium will come in. This will cause a self-propagating action potential to be generated. Okay? Now, my self-propagating action potential is going to continue down my sarcolemma. All right? Now, the sarcolemma is going to go over here. Let's see. This is going to invaginate. Okay? And I'm going to erase some of this so I have more room to write. Okay? My action potential is going to travel down here and it's going to go down this structure. Okay? That structure, we're calling that T-tubule. Okay? Now, inside here are all of my myofibrils. And I got lots and lots of myofibrils. Okay? And they're coming down here, and they're doing this, and they're doing this, and they're doing this. And remember, the T-tubule is going to be wrapping around the sarcomeres. Now, around these things, I also, and I know this is a terrible drawing, how I'm doing this, but that is representing the sarcoplasmic reticulum. Okay? Now, on the sarcoplasmic reticulum, actually, let me do this. Oh, I need a different color. I'm going to have, in yellow, calcium voltage gates. Okay? The action potential, let's change the action potential color to black, is going to come down here, and it's going to allow sodium into here. All right, that is also going to change the voltage in this area. And when I change the voltage here, I need a new color. Let me see if I can do this. Then I'm going to have calcium molecules that come out. All right, so this is representing my calcium molecules. Those are going to diffuse to my sliding filament mechanism. Now with my sliding filament mechanism, I can draw right here my myosin, and then right here I can draw my actin. Okay, this is the Z disc, this is the Z disc. So this whole thing is a sarcomere. Okay. And then the calcium is basically going to start coming down here, and it's going towards the troponin. All right, let's blow up the troponin. So if I have, ooh, I like that. My actin molecules right here. Okay, and then my actin molecules have attachment sites on them. Okay, but they also have troponin blocking it, and they have tropomyosin blocking it. So the yellow is troponin, and the black is tropomyosin. And this is the actin. Okay, I want to get one of these heads on. All right, and remember they have an ADP and a P molecule on it. I want to get that to attach to this. So the calcium is going to come over here, and it's going to change the shape of the troponin and tropomyosin so that they move, exposing the actin right there. Okay? Now, what I can do is show that my myosin head is then going to attach to this. All right? And it's going to attach, and then it's going to go to this form. All right, so we're going to do this. And it's going to swivel forward, pulling, let me erase this, the actin. So you go from this over here, and it pulls the actin towards the midline. So it's going to pull it in that direction. Okay? Now, when you do that, the ADP and the P float away. Oops. Okay. Now, I still need this to recock. Okay, I need it to detach and recock. So what's going to happen is I have over here, oops, I want a different color. I have over here ATP, okay? The ATP is going to come over here and attach to that. When it does attach it, then this will pop off, and I will have my ATP molecule attached to it, but this will still be in the uncocked position. Okay, so I'm going to hydrolyze the ATP. So it's like setting off a little bomb underneath that, which causes this to go into the upright position. And that will then take you back to there, and you will have the loose ADP and P. Okay? Now, the one thing I have not mentioned as we've gone through this is these calcium molecules. As soon as they are released So one, they get released from the sarcoplasmic reticulum, but remember they're toxic. So as soon as they're released, you're going to start vacuuming them back up into the sarcoplasmic reticulum so that they aren't hanging around doing damage, okay? All right, I think you've got an idea now of how you can draw this out and practice it. And you can see that you don't have to have great artwork. The important part of doing this isn't that it looks like a Van Gogh, it's that you have gone through this process tactily and visually, and hopefully you're explaining it out loud as you're going about it, okay? All right, we're gonna stop now, and I'm gonna end this recording.